All right, so in the first part of cellular metabolism, we basically took our glucose, we broke it down, and we were able to make ATP out of it. Right? With anaerobic, we can just stuck with glycolysis in the lactic acid pathway. We don't need oxygen for that. But if we do aerobic, we have oxygen available. We can go to the citric acid cycle and go to the electron transport chain, and we can do our oxidative phosphorylation there with our ATP synthase, and we can get that big payout of ATP. Okay, but remember I said if ATP's available, all right, if there's lots of ATP available, we don't need to be doing glycolysis. We don't make these stores a big pool of ATP because basically ATP will revert back to ADP. And basically we broke down all this glucose, used all this energy to make this ATP, and then it's useless to us. All right, so depending on what's happening in the body depends on where the glucose gets shunted or the nutrients get shunted. So that's what we're going to kind of discuss in this second part of metabolism. We're going to talk about where glucose goes, what's the pathways, where can we send it to, depending on what the energy needs are in the body. We'll also talk about our breaking down lipids, breaking down fats, those fat stores, and then also looking at amino acid metabolism. The three big, basic macronutrients are glucose or carbohydrates, our lipids, and our amino acids or our proteins. So when energy levels are high, we have enough ATP around, we're resting, and we've already ate a meal, and so we have all this glucose running around if we carbohydrates in our meal. So one way the body can store glucose is as glycogen. Okay, it's just all these long branching molecules of carbohydrates. You can see here glycogenin, that is this protein here. And all we're going to do is string these long molecules of carbohydrates. You're just building in these carbohydrates, making these strings. Okay, so now we have the store of glucose in the cells that we're going to be in is the skeletal muscles, the liver, okay, are our two main stores of glycogen. We'll see the skeletal muscles, they can have the stores of glycogen so they can break it down to use as glucose for themselves. Whereas the liver can break down glycogen and put glucose out into the blood. Okay, so this is one store. Basically, we can keep about 120 grams of glycogen in our liver. So we have a store, but it's not a grand store, so it can get depleted fairly easily if you're doing, say, strenuous exercise or you're fasting for a long period of time. So two terms you should know, glycogenesis and glycogenolysis. Okay, glycogenesis is the making of glucose. When we eat a big bag of Skittles, we're resting, we're not using glucose. One of the ways we can store it away is by forming glycogen. Our hormone insulin induces this. It causes the liver, causes the skeletal muscle to take up glucose, and it also promotes the formation of glycogen. So if our glycogen stores are down, we can store some of the glucose, or our glucose stores are down, we can store it away as glycogen. But there is a limit to what we can store. We can't just keep making glycogen, keep making glycogen. There is a limit in which the cells of the liver and the skeletal muscle have in making glycogen. Okay, we'll see what other store do we have. We have fat. We'll get to that in a bit. But if we stored away enough glycogen, we've reached our maximum, then we got to go to that other pathway. But say we hadn't been eating for a long period of time, and then we put sugar in, we're going to use that sugar for some energy, but we can also store some away from glycogen if our glycogen stores have been depleted. Okay, so this is kind of the intermingling of the metabolism. If something's going different in the body, where are we shunting the nutrients to? Are we using it to make ATP? We're shunting it away to glycogen, or we're going to shun it away to other areas. Okay, on the other opposite end is glycogenolysis. That is the breakdown of glycogen. Now we can break down, if we need glucose, we can break down glycogen to produce that glucose. So glycogenolysis operates in breaking down glycogen, depending on the energy needs. If you're in a fasting state, right, your blood sugar goes down, your cells are getting less glucose, you're going to want to increase blood glucose, and one way to do this is glycogenolysis, chiefly through the liver. But let's look at kind of the fate here of glucose. Here's our glucose in our blood. We eat our bag of Skittles. That glucose can go into many tissues. All right, the tissues can use it as glycolysis. Say they need energy. There's not enough ATP. They can shunt that through and go to glycolysis, and if oxygen is available, we can do our aerobic respiration and get our ATP. But what if energy needs are met in the cell? Chiefly the skeletal muscle, the liver, are the two big components in which glycogen gets made. We can shunt the glucose up this way and produce our glycogen. Okay, do our glycogenesis, making glycogen. All right, so now we have our stores of glycogen. But what happens if we go in the opposite direction? Now blood sugar's going down, the cells are depleted. Chiefly our skeletal muscle and our livers are going to be at play in the glycogen. We can break down glycogen, and then the cells can use it for glycolysis. Okay. One thing we did say is that the liver is the big store of glycogen. And the liver is the only one that can do this. You're going to see the liver is a big player in these interconversion of nutrients through the body. But the liver can do breakdown of glycogen and release the glucose out into the blood. Okay, so the muscles, they have big stores of glycogen, but they're going to use it for their own needs. They're selfish and they're going to be using glycogen, break it down to glucose to go use it for glycolysis. The liver, on the other hand, is a giver and can break down glycogen, and because it has the enzymes to remove this phosphate, we're able to put the glucose back into the blood for other cells to use if glucose levels are low in the body. Okay, so that's kind of the movement. Where do we shunt glucose? Okay, energy needs are met, we can shunt it through, make glycogen. Energy needs aren't met, glucose is low in the blood, the liver can break it down and send it out to the body so the cells have glucose to use to make energy. All right, so that gets us through glucose metabolism for what we need. So now we're going to move into metabolism of lipids, right? Nice belly here, everybody's favorite, lipids, and metabolism of proteins. Okay, because these are our three major macronutrients. We've got our carbohydrates, our lipids, and our proteins. So besides the glucose, we can use lipids and proteins for energy. Usually proteins are one of the last things because we want to maintain our proteins for our lean muscle mass. Okay, but we're going to be using the same pathways. They're going to enter. How do we get ATP? Because ATP is our energy. How do we get ATP out of these other macronutrients? They're going to go through the same pathways as pyruvic acid would go. We're going to see the unifying molecule for all of this is going to be our acetyl-CoA. And we'll see this in a sec when we start looking at the pathway for metabolism for these nutrients. Okay, so depending on what the needs are, say we've been fasting or we've been working out and our glucose is down, our glycogen has been depleted. Now we can start breaking into our lipid stores. Okay, but what about if we eat a bunch of Skittles, or we eat a large meals, we're not resting, our glycogen stores are met, we can store more away as lipids. So we'll first walk through our lipid metabolism, look at when do we store as lipids, and then look at the actual breakdown of lipids and how we get ATP from breaking down our lipids. So here is our production, lipogenesis, making of lipids. This would be called lipogenesis. And so our energy stores are met. We have enough ATP, but we still have glucose, or we still have nutrients coming in. Our glucose can go down our glycolysis pathway. But now, instead of going in, we already have ATP levels met, so we don't need to be going through the citric acid cycle to make more ATP. The products of this pathway can get shunted into making lipids, making fats. And if you remember from our chemistry days, we have a backbone of glycerol. Right? It's that 3-carbon backbone, and then we put fatty acids, 3 fatty acids here. Okay? Remember, these fatty acids, they are just long chains of carbons. So these products of glycolysis, instead of going into the citric acid cycle, because we already got the ATP needs met, we've got glycogen stored away, we can store it away as fat. Okay, so this product, glycolysis, used to make the backbone, 3-carbon backbone, and then acetyl-CoA. If you remember acetyl-CoA, it was a 2-carbon molecule. Okay, what we're going to do to make our fatty acids is essentially take acetyl-CoA and keep adding acetyl-CoA. So we keep adding 2 carbons to this chain here to make our fatty acids for store. Right? So typically our fatty acids are an even number of carbons. There are some odd, but most are even number of carbons. But if we make it this way, what do you see if we need to go the reverse direction? We need to go the reverse direction. We need energy. What's the obvious choice? Let's break down those fatty acids to acetyl-CoA, and what can we do with acetyl-CoA? We can bring acetyl-CoA into the citric acid cycle, and by doing that, we can convert these fatty acids into ATP. Okay, so energy stores are met, glycogen is met, we can shunt and store away. Again, the 3-phosphoryls becomes our backbone, and our acetyl-CoAs, we add them together to make our long chains of fatty acids. Right? So this side just basically expresses what I expressed on the previous slide. What happens to the glucose that's going down? It gets shunted to fatty acids, and again, those fatty acids, we get the glycerol backbone, and what are we going to do? We're going to add or put those acetyl-CoA molecules together to do what we call lipogenesis. Okay? And this is going to occur in adipose tissue and the liver, where we'll see the lipogenesis. Remember, our liver is going to be basically where we interconvert all our nutrients. It is a key organ, so you want to treat your liver nice because it's involved in all this metabolism pathways. Okay. As I mentioned, kind of the unifying molecule, and we'll see this is also for our amino acid metabolism, is acetyl-CoA. Right? Acetyl-CoA can be entered into the citric acid cycle, and it can give us ATP. But depending on what the needs are, if the cell needs energy, where are we going to take it? We'll take the acetyl-CoA to the citric acid cycle to make ATP. But what if energy needs are met? All right, the acetyl-CoA can be converted into fatty acids. We can go to ketone bodies, and off to other locations. Here's that storage pathway with our fatty acids. So that's kind of our unifying molecule. We can convert amino acids to acetyl-CoA. We'll see later. We can convert fatty acids to acetyl-CoA if we need energy, or the acetyl-CoA, if we don't have an energy, we don't have to store it away, we can bring it into the citric acid cycle. So that's kind of, this is that unifying, one of the unifying molecules in metabolism. We get acetyl-CoA. If energies are met, we can store it away. If we need energy, we can break down fats, make acetyl-CoA, and if energies aren't met, the glucose that's made from acetyl-CoA will enter in the citric acid cycle. Right? So let's look at, we're here because we already did this portion. Sorry, I got so much writing here, but we did this. Now what if energy needs aren't met? We've been fasting, we don't have, for glucose getting low, we need ATP. We can go the opposite direction and break down fatty acids to acetyl-CoA, and we can send that acetyl-CoA now into the citric acid cycle. Our white adipose tissue. Everybody's favorite, right? That's that white fatty tissue, good store. You'll see, great way to store it because we can get 9 kilocalories. You're going to see we can get a big payout of ATP by breaking down fat. So it is a good storage in the fact that if we need energy, our energy is ATP, we can get a big store of fatty acids. But unlike glycogen we can only store away, say 120 grams in the liver, and then we've met our max, our fats, and we can keep building more. As you know, over the years, professors put some on, but we can keep storing that away. If we need energy though, we've made our fat. Now you start dieting and you want to get rid of the fat, you start kind of reducing your calories, so forth. Your body can do lipolysis. Lipolysis, is the breakdown of fats. We can break down the fat and now use it as ATP. We're not eating a lot, we're reducing our calories, but our cells are still demanding calories. Right? We don't have all this glucose in our blood to basically use, and we've depleted glycogen. Now we can start going after and using that fat as energy. We're going to break down, and those fatty acids we can break down in, remember when we built the fatty acids, it was acetyl-CoA, bound to acetyl-CoA, bound to acetyl-CoA, we just made these chains putting together acetyl-CoAs, these two carbons. Now we can break down to acetyl-CoA and send it into the citric acid cycle. Alright, so here we go. Let's get rid of some of that extra fat we're carrying around. And so to do that, we got to break down our fatty acids, use them for energy source. And how we do this, it's called beta oxidation. And what we're going to be doing, if you remember, these fatty acids are put together, right? We have the glycerol backbone, the 3 carbon that holds the 3 fatty acids. The fatty acids were put together. We basically bound acetyl-CoAs. All right, we were putting those 2 carbons together, building up that fatty acids. So most of these fatty acids are even number, 14, 16, 18. So we break them down. When we go to break them down, we're going to form acetyl-CoAs. All right, and if you remember, the acetyl-CoAs we can use in the citric acid cycle. We go acetyl-CoA through the citric acid cycle, and we get those NADH to FADH2, and we bring that to the electron transport chain, and we're able to make our ATP through oxidative phosphorylation. All right, so here's the process. The fatty acids. Remember, these fatty acids are large chains of carbon. This is just showing the end. For example, I'm going to use a 16 carbon fatty acid, right? And if you remember, each acetyl-CoA is a 2 carbon molecule. So how many do you think we can make from 16? We can make 8 acetyl-CoA, right? This beta oxidation here, and we're oxidizing, so something has to be reduced. This process here is cleaving off the acetyl-CoA. So this is just showing the end of the fatty acid. If this is a 16 carbon fatty acid, here's 3, and there would be 13 more carbons down the way here. This is just showing the end and what's happening molecularly. Okay, what I want you to notice along the way, so this is just going to cleave off 2 of the carbons, right? So we're actually, for a 16 carbon fatty acid, we're actually going to do this 7 times. Okay, and each time we cleave off the acetyl-CoA, we're reducing a molecule of FADH2, which we can use at the electron transport chain, and reducing a molecule of NAD. And if you remember, the 1.5 this is how much that's worth at the electron transport chain. The 2.5 here is how much the NADH. So we're going to get ATP here, or the ability to make ATP, by 2 ways here. By the breakdown of the fatty acids, but when we break it down, remember, we form acetyl-CoA. So we'll get ATP from this process here, breaking down, but then we can also obtain ATP by bringing that acetyl-CoA we form into the citric acid cycle. Okay, and you can see, if you remember, acetyl-CoA, just 1 molecule of acetyl-CoA going through the citric acid cycle is worth, right? We produce or reduce 3 molecules of NAD, 1 molecule of FAD, and produce 1 ATP. Okay. Those were the numbers from 1 acetyl-CoA going through the citric acid cycle. How much is this worth? Right? 2.5 ATP for each one, so that's 7.5 ATP. One FADH2, that's worth 1.5. And 1 ATP. So for each acetyl-CoA that we cleave off from a fatty acid, we can get 10 ATP. All right, that's that number there. Okay, what did we say? All right, so we're going to form 8 acetyl-CoA. And how much is each of that worth at the electron transport chain, or through the citric acid cycle? We got 10 ATP, so 8 times 10, that's 80. Through the beta oxidation, that's the reactions here, the beta oxidation. We're able to get, because we're reducing these molecules, roughly we can form a total of 4 ATP here. All right, So this is, we have to oxidize the ends to make this happen. What you got to recognize is we're forming 8 acetyl-CoAs, but the last end here, there is no N to oxidize. All right, there's nothing there. So how many cleavages we're actually doing is we're going to do 7 cleavages. So think of it this way. Here's my carbon. All right, we're cleaving there, cleaving there, cleaving there, cleaving there, cleaving there, cleaving there, cleaving there. We don't have anything to cleave on this end. So how many we end up with? 1, 2, 3, 4, 5, 6, 7 cleavage. So that is where we're getting this 28. All right, 4, and we're cleaving 7 times. We're doing this reaction 7 times to make our 8 acetyl-CoA, and therefore 7 times 4, we get 28 ATP, right? And that gives us roughly a 108 ATP. What do we get from breaking down glucose? We got about 3 ATP, or 32 ATP. So roughly we're getting almost 3 times the amount of ATP from each fatty acid that we break down. If it's a 16 carbon fatty acid, right? There can be 14 carbons just depending on which, but now you can figure out, if I gave you a 14 carbon fatty acid, you could figure out the amounts, right? Okay, because you would know how many you form from cleaving it and how many acetyl-CoAs, and you know that the acetyl-CoAs are worth 10 ATP, and you can give me those numbers. All right, but you can see how this is a good energy store because we're able to make lots of ATP from it by breaking it down. So we're able to store lots of energy in those fatty acids that we store away. Okay, that's why sometimes they're hard to get off, because we got to burn a lot of energy to make them go away. All right, so just a general rundown of the cleavage. All right, again, as we beta oxidize, we're forming 2.5 ATP and 1.5 ATP from those that we're doing through the beta oxidation reaction. All right, let's look at another fuel the body can use, ketone bodies. Okay, ketone bodies, we form of acetone, azoacetic acid, and beta hydroxybutyric acid. Okay. This is kind of the premise of the ketogenic diet. Is what people measure are measuring how much of these ketones. And why might this be important? Because, right, lipolysis. If we break down fat faster than we are actually utilizing it, okay, as in dieting, the ketogenic diet makes us break down fat faster than we're utilizing it. Starvation, diabetes are ways in which this is not a good way, but the ketogenic diet is basically to induce fat breakdown. But if you're not utilizing it, it's staying in the bloodstream, and what the liver will do will convert those fatty acids that are in the bloodstream, okay, to acetyl-CoA and then to ketone bodies. Key is this is a source of energy. Your cells can use ketone bodies to form back to acetyl-CoA. And where can we use acetyl-CoA? Yeah, we can bring it into the citric acid cycle to make energy. So your body can be, many of your cells can use ketones as fuel to make ATP. Some cells require glucose, but many of the cells can use alternative fuels. One of those is your ketone bodies. Okay. If you see, is acidic. So if you were doing a ketogenic diet, what do you think your blood do you think your blood would be more on the basic side or starting to over more on the acidic side? Yeah, since ketones are acidic, if you're making lots of ketones, you're making your blood a little more acidic. You're not going to make it pathologically acidic like you would with diabetes, but it might be more on the line of the keto end or the acidic end of that homeostatic range if you're producing lots of ketones. Okay, but again, it is a fuel because we can convert them to acetyl-CoA and use that in the citric acid cycle and make our ATP through this way. If we're using acetyl-CoA though for energy, what must we have available? Yeah, we have to have the oxygen available because cannot use a citric acid cycle unless there is oxygen available. All right, so lastly, we'll look at amino acid metabolism. Okay, some of the things, amino acids, we have amino groups, so it provides nitrogen for the body. Okay, dietary proteins, that allows us to build up muscles. Okay, this is the last line of energy source. We want to conserve our lean muscles because we need it for movements and to move and so forth. So it is the last. Use glucose. Glucose not available. We can use fatty acids. Fatty acids break down. We can use ketones. Amino acids we want to save because they're used for our proteins. But if, say, we're in starvation mode, you're going to start breaking down amino acids, and you can use them to make ATP if need be. Okay. Our bodies, okay, we have essential and non-essential amino acids, and you'll see why in a second. We can actually make amino acids from other amino acids. So there are 12 of these, and in kids there's only 11, but there are 12 that are considered non-essential. We have the enzymes to make these amino acids. There are 8 of them in which we have to get to the diet. If we don't get them in the diet, we don't have them, and we can't build whatever proteins are needed that require those other 8 amino acids. Okay, because we cannot make those amino acids. These other 12s, we have molecules in our body we can convert to make these amino acids. All right. So that is the difference between essential and non-essential. Non-essential we can make. All right, we have the enzymes to make it. The 8 essential, we don't have the enzymes, and you have to get those through your diet. Here's the rundown of them. Okay, histidine. All right. Until histidine's over here for adults. It is non-essential, but for children, you have to get histidine in the diet. Okay. How can we make these amino acids? We use a reaction called transamination. And what molecules do we use to make these non-essential amino acids? We will see. I didn't have you keep the names, but I want you to realize that some of the building blocks for our essential amino acids lie in those intermediates in the citric acid cycle. Okay, many of the molecules that we're going to make our non-essential amino acids are products, intermediates in that citric acid cycle or pyruvic acid. All we're going to be doing is adding an amine group to them. Okay, we're going to take an amine group off of a donor amino acid and we're going to add it to one of these molecules and we can make our non-essential amino acids. Okay, we can we're going to see glutamic acid is going to be a good donor for our amine. We got to get our amine group from somewhere. And we'll see through the transamination, we take it off one amino acid and put it onto one of these molecules, and we can get our non-essential amino acids. Okay. Each transamination, so each of those non-essential amino acids, we have to have specific enzymes for the reaction. So we have to have at least 12 different enzymes to make the 12 different non-essential amino acids. Okay, and requires vitamins as coenzyme, so make sure you get your vitamins in your diet, because one of those, B6, is a coenzyme for these transamination reactions. So let's make our non-essential amino acids here. We're going to do this through what's called transamination. Basically, we're swapping an amine group from what our donor is here, glutamic acid, to one of those intermediates in citric acid cycle, right? Oxaloacetic acid. Hopefully that looks familiar. Oxaloacetic acid is that molecule that binds with acetyl-CoA to start the forming citric acid to start the citric acid cycle. And so basically, what are we doing in these transamination? We're swapping this bond there. When we add an amine, okay, again, glutamic acid is our donor of our amine group. We're adding an amine to oxaloacetic acid, we form aspartic acid. Okay, one of our non-essential amino acids. Here, another one of our cellular respiration intermediates, pyruvic acid. Again, we got glutamic acid, donating the amine group. We're swapping there to there. And when we add the amine to pyruvic acid, we create alanine, another non-essential amino acids. You see AST, ALT, different enzymes for these reactions. We'll have different enzymes to do this for creating other non-essential amino acids. Okay, but you can see how we're able to form these non-essential amino acids out of non-amino acid molecules. Okay, unfortunately, essential, we got to get in the diet. We don't have an enzyme that can swap to another molecule to make us an amino acid. Right, so looking at amino acids as energy. Okay, you can see if I strip some of my amino acids of my amine group, I can make them into, say, pyruvic acid, lysine and tryptophan, and if I strip the amino acids, I can ultimately make them into acetyl-CoA. And so you can see how this amino acids, if I can convert these to acetyl-CoA, I can ultimately use these to make ATP. Okay, it's not as efficient as using glucose or lipids, so it's not a great, hey, let's use amino acids as our energy source. We get a more bang for a buck when we go through our glucose and breakdown, and also lipid breakdowns. We're able to convert those. Okay, but you can also see is, say you ate a diet of all amino acids. Amino acids are great for protein, but what if we have too much amino acids? We've made enough energy to fuel our cells, and we have this glutton. We ate all this protein. We ate a bunch of protein shakes. We still have lots of calories above what we're going to burn. Okay, the amino acids can be converted to shunted to basically fat, right? Because acetyl-CoAs are building blocks for our fatty acids. So you can see kind of this intertwinedness between these metabolic pathways. Okay, this is the key molecule though. This one we can shunt in many different directions. We can use it in the citric acid cycle. We can also, if we need to make an amino acids, we can go the opposite direction and make amino acids, and we can also shunt it away to make fat. We can store it away in fatty acids. Okay, so